So we welcome Madina Wane, who did her bachelor's in biochemistry at Imperial, then an MRes in molecular and cellular basis of infection and immunity at Imperial, and she is in about 2 or 3 weeks about to defend her PhD thesis in immunology at Imperial. She's also co-founder of Black and Immuno, which works to celebrate, amplify and support Black voices in immunity. And today she'll be talking us through some of the work that she did in her PhD. Thanks, Tina.
Cool. So thanks so much for the introduction and to the organisers for inviting me. I'm really excited to share my talk, kind of outside of just immunologists today. So as was mentioned, I'm just finishing up my PhD in immunology, and today I'm going to try and talk to you a little bit about my research, but also because I know that a lot of you will be from other fields, chemistry and so on, hopefully try and make it applicable and share some of my kind of research process and approaches to problem solving, both for my scientific research, but also for some of my projects to tackle inequality in science, so kind of more social issues. Okay, cool. So I'm going to start with discussing a little bit of like the wider context of why it's important to think about science in the larger context of society. And then I'll talk about my specific PhD research, which was on respiratory disease. And then I'll focus more on my projects addressing inequality in science. So I think in the last 12 months, all of us have been kind of hyper aware of how important science is in tackling some of the problems that society is facing. Without the amazing science that a lot of people have developed over the last year, we would have struggled even more to tackle what's kind of one of the biggest health crises in modern history. So we know how big of an impact science can have, but I think something that's really important to me and really important to consider is that we can't just think of science on its own. We have to think about the wider context that drives scientific research, allows it to continue, and what are some of the factors that can kind of impair that process. So, for example, thinking about how are scientists themselves supported, what's the infrastructure, what are the positive and negative impacts on scientists, and how can we make things better for the people that are working in this community. So I kind of want to raise the point during this talk that I'll be talking about my own interests and my own passions, but really I think it would be great if you can think about the things that you're interested in. Maybe there's some scientific questions that you're particularly interested in, or there's some aspects in your community, your local community, or beyond that, you'd like to make a change or make a contribution to. And hopefully by the end of this talk, you'll kind of have a little bit of insight in the ways that you can tackle some of the issues that you're passionate about. Okay, so I'll move on to talking about my PhD research. And as was already mentioned in the introduction, I did my undergraduate in biochemistry, which is a pretty broad subject. Maybe some of the chemists do some biochemistry modules, I don't know. But I then specialized in infection and immunity. And I've always been interested in infection and infectious disease, partly because of the impact that infectious disease has on the world, but also because it's a really complicated and kind of always changing topic. So, even now with COVID, we're seeing a new virus that has emerged that scientists are having to tackle. So you're kind of never short of a challenge to address. And in my research, my PhD research, I focus on respiratory diseases. So again, obviously very relevant topic, so I won't go into too much detail because I think everyone is aware of the some of the impact that respiratory diseases have, but just to highlight that there's different causes and factors that influence respiratory disease. So you have your pathogens, things like viruses, which cause infections, but you also have things like chronic inflammatory conditions, so things like asthma, which we're still struggling to understand the causes of that. And then you also have respiratory diseases that are caused by pollutants or toxins, things like cigarette smoke, for example. And all of these kind of combine to create some of the kind of leading causes of illness and death in the world. So this is obviously a really huge problem. So to try and tackle it, we need to break it down into smaller chunks. And I'm an immunologist, so I'm interested in the immune system, and there's several specific questions that immunologists are still working on to better understand respiratory diseases. So one of them is understanding, in terms of viral infection, which is the side that I was focused on, understanding what the immune response is like at the site of infections. So that's in your respiratory system, from your nose to your lungs, understanding what happens when the virus interacts with your immune system there. And for a lot of research, especially in humans, when we study the immune response to respiratory viruses, we often look in the blood because that's really easily accessible to get a blood sample for someone, compared to having to perform some kind of invasive procedure to get access to their lungs. So we can get some idea about the immune response, but that doesn't necessarily correlate to the immune response in the respiratory system. So that's a big area that immunologists are trying to understand. Another question is understanding what are the factors that lead to a protective immune response. So normally when we think about the immune system, we think about it in terms of protecting from infection, but another important aspect is if the immune response isn't well controlled or regulated, or if you have the wrong type of response, that can actually lead to damage and more disease. So one of the best examples for this is the flu. So when you get the flu, normally the period when you're most sick is when most of the virus has already been cleared from your system. And actually what's causing a lot of the symptoms is your immune system overreacting and causing kind of tissue damage and other negative impacts. So understanding what are the factors that lead to a more protective rather than damaging response is really important. And all of these questions really are so we can develop treatments, vaccines, therapeutics that are effective. It's actually really impressive that there are now 3 COVID vaccines available, at least 3 COVID vaccines available, because for most respiratory viral infections, there's not many options for vaccines or for treatments. And when they are, they're not always that effective. So I think actually that goes to show that if you can have really supported science in terms of funding, in terms of infrastructure, you actually can make a really big impact to some of the biggest problems that we've been trying to tackle for a long time. So when we're trying to address these questions in respiratory disease research, we need to look at the different tools that we've got available. And like most biological research, scientists tend to use either cells in a dish that have been taken from humans or animals, known as in vitro research, or we can look at animals themselves to be able to study a more complete biological system, or we can also look at humans and perform clinical studies. And obviously we have to do that when we're developing therapeutics, perform clinical trials. So all of these different tools have their own benefits, but also limitations. So for example, using cells in a dish, performing in vitro research is probably one of the cheaper options. But when you're studying something like the immune system, you really need to be able to address a complete system that has a variety of different immune cells, because that's really important for the immune response. And another important aspect of the immune system is these different tissue structures within the body. And when you're performing research in a dish, you don't necessarily have access to that. So that's why a lot of people also work in animals, but obviously they are more expensive. That also have a lot of biological differences to humans. Obviously they're not completely the same. And then with humans, you'd think that was kind of like the best model or best tool to use, but obviously there are loads of ethical considerations, important ethical barriers, and also it's quite complicated and very expensive to perform clinical studies. So actually what you want is to be able to have a larger toolkit so you have more options to address the specific research question that you're interested in. So the group where I did or am finishing my PhD, the Dalman group at Imperial, focuses on using zebrafish, which is a small tropical fish. It's about, I think, 4 to 5 centimeters at adult stage, so they're really small, cheap and easy to keep. And the Dalman group uses this fish to study different aspects of immunology. And in my case, because I'm interested in respiratory disease, I focus on using the zebrafish gills, which is their equivalent to the lungs. It's a tissue where they perform gas exchange. So to give a bit more context about why zebrafish can be a useful model in biology research, and more specifically viral infection research, we kind of need to look at 2 aspects. So one is their biology. So obviously zebrafish look very different to humans. They live in water, they don't breathe air, etc., etc. But a lot of the fundamental aspects of biology is still quite similar. So for example, they share about 70% of genes with humans. And in terms of their immune system, they have a lot of the similar immune cells, which produce, again, similar immune molecules, which are used to fight off infection. And they're also susceptible to viral infection. And some researchers have started using human viruses as well, although it's still a pretty kind of new field or new area in zebrafish research. And then the other aspect to understanding why we might use zebrafish as a model is to look at the tools available to perform different studies. So I already mentioned zebrafish are kind of small, they're cheap, easy to keep, they breed a lot as well. Another aspect is that when they are in the early stages of development, they are transparent, so they don't have any pigment. And so that means it's really easy to study them with microscopy to visualize some of the biological processes. There are also zebrafish mutants, which lack pigments at older stages. So again, you can see inside the animal and visualize some of these biological processes in a living animal, which is really difficult to do in animals like mice, for example. We've also got lots of tools to be able to make genetic modifications to animals, and this allows us to do things like label different cells of interest, so that again, we can visualize them with different tools like microscopy. So to kind of put this into the context of respiratory disease research, my PhD was focused on evaluating the zebrafish gill specifically as a model to study respiratory immunology, so understanding the immune system. And really, I wanted to evaluate, can the gills be used as a model, and what would they be useful for? So to answer that, I kind of split it up into 2 main chunks. And I think this is generally a good approach when you're trying to evaluate systems, not necessarily in biology, but other fields as well. So one is to understand the kind of basic aspects of your system. So for me, that was understanding the fundamental biology and immunology of the zebrafish gills. A lot of research was focused on their respiratory function, but not so much on their immune function. So for me, I wanted to expand that area of the literature. So then we could compare to other systems or other animals and species in terms of their immune system. And then the second aspect is testing the model in different contexts to understand how it can be used or what it can be used to study. And again, remembering that my specific interest is in respiratory viruses. Okay, so one aspect of the immune system that's really important for fighting infection are these specialized structures known as lymphoid tissues. So these lymphoid tissues are basically contain these accumulations of specialized immune cells known as lymphocytes. Lymphocytes are cells, there are lots of different types. Some of their main functions are things like making antibodies against particular antigens and also fighting off infected cells. So you have different types of lymphoid tissues in mammals. One of them are lymph nodes, which I think most people are probably familiar with. Again, these structures contain these accumulations of lymphocytes, and they're found throughout your body. You might find them kind of under your neck. If you get an infection, often you can feel they're quite swollen. And that's because the cells have become activated and have proliferated, so they're kind of much larger numbers of cells in those structures during an infection. You also have lymphoid tissues in mucosal tissues, and mucosal tissues are tissues that are exposed to the external environment and covered with a mucous barrier. So, for example, in your nose, in your lungs, in your gut. And again, the lymphoid tissues in those tissues become activated when they're exposed to kind of foreign material pathogens, and that allows an immune response that allows the cells to respond to infection or kind of other materials. So in fish, so far there haven't been any lymph nodes identified. However, there have been mucosal associated lymphoid tissues identified in various types of tissues. And there's been a structure of an accumulation of lymphocytes identified in salmon and trout gills and some other fish species as well. But it's not quite clear whether this structure or how similar the structure is to mammalian lymphoid tissues. So that's something that I wanted to address in my PhD. And to do that, I used these genetically modified fish which produce a fluorescent protein in a certain cell of interest. In my case, this was in a population of lymphocytes. So I could take the gills from these fish, and then I could use antibodies that would recognize the fluorescent protein, and then visualize this under the microscope, well, with confocal microscopy. So here I'm just showing an image of a zebrafish gill at 21 days. So this is still when the fish is pretty young, a juvenile stage. And in gray is just the structure, this kind of highly branched structure of the gills. And in blue, these are lymphocytes scattered around the tissue. You can see it's not really got any consistent pattern. And when I look just 9 days later, you can see that the blue lymphocyte cells are almost exclusively located in this top region of the gill. So forming these kind of accumulations, which I showed previously. So that seemed to indicate that, okay, in zebrafish, we probably do have a similar lymphoid accumulation in the gills, but now we need to understand a bit more what the structure is and how it compares to mammalian lymphoid tissues. So one of the important aspects of lymphoid tissues in mammals is that they're not just made up of one type of cell, they're made up of lots of different types of cells because each cell has a different function. And in order to get an immune response, you need to be able to have these cells interacting with each other. Another aspect that's important for the immune response is this, again, this dynamic system where different cells can interact with each other. And in the images I'm showing here, because this is from fixed tissue that's been taken from an animal, it's just a snapshot of the cell, so you can't actually see how the cells behave. So to address that, what I did was apply a technique that's used in other models where you can take a tissue from an animal and you can keep it in a specialized solution to keep the tissue alive for some time. Sometimes it's just a few hours, but it can go for days to even 2 weeks in some cases. So again, using genetically modified fish, which produce these fluorescent proteins in different cell types, and then imaging these gills with confocal microscopy, I was able to take videos of the gill tissue to see how the cells behave. So I'm gonna, let's see if I can play this video. This might be a PowerPoint issue. Okay, well, unfortunately, I don't think I can play the video, but I'll show you in the next slide, like kind of what we can learn from it. But you can see, at least in the image, that there are two different colored cells representing two types of immune cells. So in blue, you've got these B cells, which are lymphocytes that usually produce antibodies. And then you've got another type of immune cell, in this case, CD4 positive cells. some of these will be a different type of lymphocyte cell, and some of these will be a completely different immune cell altogether, known as myeloid cells. So at least you can see we've got the presence of different type of immune cells within this lymphoid structure that I previously described. So to look at more what the cells might be doing in this tissue, I wanted to look at how they interact with foreign material, which again is a really important aspect of the immune response, because immune cells need to be able to detect foreign material and then respond to it if it's, looks like it's going to be harmful to the host. So to do that, I used a protein which was conjugated to a fluorescent compound. And in this case, this is just something that I bought, is commercially available, but in other aspects of my PhD research, or in the Dauman group, we've also collaborated with a chemist who was able to synthesize a kind of compound of interest that we wanted to test and conjugate it to a fluorescent compound. So there's definitely room for a lot of interdisciplinary collaboration, especially because zebrafish, because it's still a fairly new model, it's only really been used in biology since like the 70s. There's still a lot of reagents and tools that aren't necessarily available like they are for mice, for example. So it's a good opportunity to be able to kind of innovate and work between fields to develop new tools and new reagents. Okay, so for this experiment, I applied this fluorescent protein to the gills of the fish. And then after some time, I took out the gills and again visualized them by confocal microscopy to see what the cells were doing. So if you focus on this pink arrow, it's pointing to this yellow cell, this big yellow immune cell that has taken up some of this fluorescent protein. So already I can see that these cells are capable of interacting with foreign material. And I can track them over time to see how they move. And if you follow this to the last point, so this is around 17 minutes, you can see that this yellow cell that has taken up the protein is also interacting with another type of immune cell, this B cell. And these kind of behaviors are things that we see in mammalian lymphoid tissues. So this was giving an indication that in terms of the biology of zebrafish gills, there are some kind of core similarities that we see in mammalian species. Obviously, there needs to be more work to look at more of the kind of specific functions and the consequences of some of these cell behaviors, but it was giving us an initial indication that maybe they could be used to study similar processes. So the other aspect of my PhD was testing the model, like I mentioned earlier, and I actually started this project during my master's, working with the same group, with the Dauman group, and we worked on a collaborative project with researchers that work on mice and researchers that work on clinical studies at Imperial as well, looking at respiratory viruses. And what we wanted to do was compare the immune response across these species. So to do that, we used a compound that kind of mimics some aspect, it's kind of viral-like compound. It mimics some aspect of like viral genetic material. And when we applied this compound to the gills or to like mouse and human noses, we could see that all three species responded by kind of switching on a gene that's important in antiviral responses within a few hours. You can see they've got really similar pattern of increase in this particular gene. And then we looked at a different compound, which again mimics some aspects of viral genetic material, but it stimulates or activates cells through a different cell pathway. And when we looked at this compound, we found that zebrafish and humans didn't respond, but mice still had a pretty clear response. So this was really interesting for us because it showed us that in some contexts, zebrafish have a more similar response with humans than mice do. So to me, this really highlighted both the potential of zebrafish gills to be used as a model for respiratory and viral infection research, but also the idea that there's kind of no one model which is perfect for one field. You really need to be able to look at different species, especially in biology, to get like a more complete picture of fundamental fundamental aspects of biology, of immunology, rather than just focusing on one species. So as I mentioned, I was particularly interested in respiratory viruses. So rather than just using these kind of synthetic compounds, I wanted to see whether I could actually infect zebrafish gills to study their immune response to a true infection. So again, this was a project that involved a lot of collaboration. I worked with researchers at Imperial, with Professor Wendy Barclay's lab, who is specialized in influenza research, and also with some researchers who work on kind of fish immunity to get this fish virus, IPN virus. And this allowed me to perform some experiments where I injected zebrafish with these viruses or with a kind of control substance. And then I was able to analyze their response, looking at their gene expression. So the first thing I looked at is zebrafish immune genes and see how they responded to these different viruses. So with influenza virus, I'm just showing two genes involved in the immune response, and hopefully you can see initially there's like a small increase. So this is 1 day after I've injected the virus, but that kind of quickly goes away after a few days. So it's a very small and kind of transient response. If I look at the fish virus, the IPN virus, actually, there doesn't really seem to be much of a change happening. If anything, there's a small decrease in one of the genes. Again, this doesn't really last, and by 6 days, you're not really seeing much change. So this was showing that there wasn't really a strong response, maybe a small response with influenza, but I wanted to understand whether there was an actual infection happening in the fish, whether the virus was replicating. And for that, again, I analyzed the genes, but for the virus. And actually, this technique is the same technique that's used for for COVID testing. It's known as quantitative PCR, qPCR, and it allows you to measure gene and kind of nucleic acids. So when I looked at the viral genes for influenza, on the left, you can see this really tall bar is my control. So that's basically how much virus I put into the fish. And on the right, these are my samples, different tissues from the fish, kind of a few days after I've injected them. And you can see that the level of virus drops dramatically within a few days. So even though there's like a small amount of virus retained initially, it doesn't really seem to be replicating because it's being rapidly lost. And then for IPN virus, which was the fish virus, I didn't detect any virus in my zebrafish samples at any point. So again, showing that either there's a very low and undetectable level of virus, or there's really just no replication happening at all. So, I mean, there's a lot of reasons why the zebrafish don't seem to be susceptible to these two viruses in particular. But what's clear from using the synthetic compounds is that they do have the kind of mechanisms to be able to respond to viral-like materials. So it's not necessarily that, they just can't respond to them whatsoever, but there might be other factors influencing, if the virus is able to get into the cells and replicate and so on. So this is just kind of like a kind of small selection of some of the work I've done, but hopefully I've been able to show you that by breaking down my question of evaluating a specific model, in my case zebrafish gills, into kind of specific aspects, I've been able to identify different areas of biology that could be studied with this model. So looking at their immune cells, their response to viral-like material, their development, and from that you can kind of build on that to expand our understanding of respiratory immunology. And obviously, in order to do that, I've also needed to use different tools. So by using and applying and developing, really, I've been able to show that we've got a toolkit to be able to investigate the different aspects of biology that I showed in the previous slide. So that's kind of it in terms of my PhD research, but I want to now focus on how I've used the similar kind of approaches to addressing problems, this time on social issues, specifically inequalities within science. So during my PhD, one of the things that I really enjoyed doing was getting involved in science outreach. This is something that actually the Dalman lab has done a lot of, and I kind of continued with some of my colleagues and some of the students in the lab. And this was really a good chance to engage with a kind of wider community about my research. And one of the kind of key things that kept coming up was the issue of representation, making sure that the diversity and representation, making sure that there were scientists of all different backgrounds that were represented in science outreach, and that reflected the community in London, in the country, and so on. And noticing that actually within science there is this lack of diversity. You don't see people of all different backgrounds equally at, in prizes, in getting professorships, and so on. So I started working on projects that were trying to address this. One of these was getting involved with Wikipedia editing to showcase the biographies of different scientists, because like in science, Wikipedia also has its biases, and there are a lot of underrepresentation of scientists from certain backgrounds. But I think one of the important things is not just to recognize this lack of diversity and representation. It also got me thinking about why you have that lack of representation, lack of diversity in the first place. And so just to note that I'm going to be talking more about issues of discrimination and racism. So I know this is a sensitive topic, so just a little warning. So yeah, when we think about why we have this issue of lack of representation and diversity, it's really about the barriers that are placed in certain groups. For example, the barriers that make science inaccessible to a lot of disabled scientists, that make environments hostile to a lot of people of color, and some of this is historical, but it perpetuates, still to this day. And so focusing on one of these issues, which is racism in science and in STEM, often when we talk about racism, we probably think about kind of harassment and like interpersonal interactions, which is important, but is more like the tip of the iceberg. So within research, another big problem is how research is conducted and how that contributes and perpetuates racism. So one really important story is the story of HeLa cells, which is one of the most widely used cells in research and biological research. And until recently, it wasn't widely known that these cells were actually taken from this African-American woman, Henrietta Lacks, in the 1950s, from a biopsy of, she had cancer. They were taken from one of her cancer biopsies, but taken without her consent and used without the consent of her family. She passed away kind of a year later, but the cells were used without the consent of her family. And still to this day, these cells are being commercially sold, for scientists to be able to do their research. But this isn't something that the family really have had an input in. Now there is some work trying to compensate the family, but since the 1950s, these cells have been sold, used without very little discussion with her family, and ultimately without consent. So another issue that again furthers racism, or kind of is a big issue for racism in science, is also how research is supported, or more specifically, who is supported. So one of the big issues is research funding. And this is just an example that I took from the Engineering and Physical Sciences Research Council in the UK. And it just shows that in blue and orange, you have a big disparity in the award rate. So this is, when you apply for grants, between white PIs and PIs of ethnic minorities. And this is kind of current data, so from the last 5 years, showing that it's not just that you have historical issues of racism, they still kind of perpetuate to this day. So I think the last 12 months of the last year have been, there's been so many changes happening in the world. One of the biggest have been a lot of kind of increased recognition and protests and speaking out against specifically anti-Black racism, fueled initially by issues of police brutality in the US, but really kind of expanding beyond that to look at wider issues within society. And I think sometimes it's really easy to think that, science is like a separate issue. How is police brutality related to scientific research in any way? But I think that's where it's important to recognize that as scientists, we're part of society. So, we exist in this structure, and any issue that involves society is also going to impact on us as individuals. And a few months ago, for example, there was a student at the University of Manchester who was quite, violently racially profiled by the security on his campus as he was making his way to his student halls. So even though this isn't, we like to think of science as just the research we do, the facts and the figures. Actually, if scientists, the people who are doing the science, can't do it without fear of discrimination, then that's going to impact the kind of science we do and impact the people who are allowed to be scientists, again, leading to that issue of lack of diversity and lack of equity, more importantly. Okay, so, when all of this was happening over the summer, I was kind of in lockdown, writing my thesis, and kind of isolated in London, weighed down by everything that was happening with the pandemic, with the protests, and really feeling a little bit helpless, but trying to work out a way that I could contribute in, some small way to my own community and specifically to my research community. So I'm not the only one who kind of had this feeling. Loads of Black scientists also felt like they wanted to do something, and actually there have been a lot of movements since the summer to produce these campaigns that showcase, amplify, and build a kind of supportive environment for Black science in loads of different fields. So I think the first one was Black Birders Week, but there have been loads since then, including the Black in Chemistry. So any chemists, feel free to check that out. The Black in Physics, basically every field that you can think of has probably had some kind of Black in STEM week with like events and social media campaigns. And so seeing all these happen, that got me thinking that it would be great to see something happen for the field of immunology. And so I basically just reached out through social media and was able to connect with some amazing other Black scientists, mostly US-based, but also some in the UK. And together we decided to, build our own Black in STEM, our Black in Immuno movement. And we work with a bunch of other kind of allies and Black scientists to create this Black in Immuno week, which was filled with events and social media campaigns, start some discussion, showcase some amazing science that Black immunologists are doing around the world. And kind of building from that, we really want to create something sustainable. So again, it goes down to the question, how can we develop tools for the problem that we're trying to tackle? So we've been thinking a lot about our mission. What are the needs of Black immunologists? Where are the gaps? What's already been done and where are the gaps? And how can we make sure that our movement is inclusive and supportive? And then similarly to, how I would think about a research problem is thinking about the resources that are available. What do we need to be able to achieve these goals? We've worked a lot with research institutions, both for financial sponsorship, but also for partnership to try and engage them in addressing inequality in their own institution and communities. And we're also establishing ourselves as a non-profit organization to make sure we've got these kind of solid foundations to be able to build on from this work so it's sustainable and long-lasting. And finally, the other part, which again, I mentioned in my research, working with others, collaborating, we want to engage with as many people as possible because we recognize that this is a movement for a community. It's not just us, like, you know, working on our own, doing what we want to do. So one of our next projects is building a hub, the Black and Immuno Hub. And this is really going to be a kind of culmination of this idea of network, bringing together different Black scientists, Black immunologists, supporting them, providing them with resources so they can thrive, and being able to interact with each other. One of the big problems of being a Black scientist in any field is that there are so few Black scientists where you work that it's really hard to be able to find others in a similar position to you. So building this kind of network, I think, will help us tackle some of those issues. So hopefully I've been able to kind of show you some of the approaches that I take to tackling different problems. And hopefully you can kind of think about what your personal scientific or, you know, kind of social interests are, and, you know, be empowered to contribute. I think the biggest piece of advice that I would give is you can absolutely contribute to any challenge that you think of, but I think the most important thing is to do it with others. None of my research work, none of the work that I've done with Black and Immuno, I could have done by myself. And there are so many people willing to help, but also that provide a different perspective. So yeah, don't be afraid to rock the boat, but don't do it alone. So finally, I just want to acknowledge everyone that's helped me with my research and in Black and Immuno. There are so many people, so I won't list them all, but my supervisors, Maggie and Laurence, have been super supportive in the last few years. Loads of colleagues in the Dalman lab that have made this work possible, and lots of collaborators that I've worked with as well. Oh, sorry, that was automatic. Like I mentioned, there's a lot of us in the Black and Immuno team also growing. So like at some point, if anybody is interested or you know anyone, feel free to reach out to us. And finally, thanks to my funders, Welcome, and I guess Imperial, my host institution as well. So that's it from me. So thank you so much for listening, and I'd love to answer any of your questions.
Thanks, Madina. That was such an interesting talk. Thanks for sharing as well some of the kind of more personal work that you've been doing with Black and Immuno. So yeah, we'll take questions now. So if we've got the chat function on, so you can either post a question in the chat, or if you just want to unmute yourself and ask a question, that's fine.
Should I stop sharing my screen, or?
It's up to you.
I'll leave it up for now in case I need to refer to any kind of previous slides.
I think we've just got a lot of compliments coming through, but no questions.
Hi, Madina. I actually have a question. It's Alexis.
Hi, Alexis.
I made it. So I will back to one of your research questions when you were looking at like the young zebrafish gills to I think it was day 21 and day 30. It looked like the gills themselves weren't really even developed until about day 30, I guess, when they're more of an adult fish. And you start seeing the immune cells also kind of organizing as the gills are organizing. So do you think that there's also a possibility that the immune cells are helping the gills develop over time? And did you happen to look past day 30 to see if there was more or less organization, I guess, in like what you might consider an old zebrafish?
Yeah, that's a really good question. Alexis is one of the Black and Immuno founders as well, everyone. But that's a really good question. And yes, you're right that like the structure of the gills is very different in that short time frame, which to me is pretty amazing. I didn't study whether the immune cells were responsible for that, but I think what it shows is that the development of the organization of the immune cells and the structure of the gills are probably linked somehow. So maybe they're kind of governed by similar factors. I did look at older stages as well, and basically you don't really see any further change. You still see this these kind of accumulations of lymphocytes in this region up until, you know, old age, elderly zebrafish. It's just that the gills kind of grow in size. But obviously that doesn't mean that the function of these cells changes, because we know from other animals that as you get older, your immune function changes. Normally it gets kind of less effective. So that could be another factor to consider.
Yeah, thank you. It was a great talk.
Thank you.
Do we have any more questions from the audience?
I have one for this slide, actually. So what was the fluorescent protein that you were using?
Yeah, sure. That's a easy one to start with. So this specific fluorescent protein was GFP, so green fluorescent protein. So in this experiment, you'd expect that because it's a fluorescent protein, you can detect it by itself, but actually what happens is when you use fixative agents to fix a tissue, sometimes the fluorescence can be affected. So that's why I use an antibody to detect the fluorescent protein rather than the fluorescence of the protein itself. But there are loads of fluorescent proteins now that are used in research. Like some of them have been developed, you know, by like protein engineering. Others are just from the kind of native species themselves. But you've got like a whole robo panel of fluorescent proteins available. Lots of them are used in zebrafish as well.
Thank you.
I've got a question, Medina, bit of a random one, but they're really beautiful images you've taken on the microscope. I was just wondering, how long does it take you to acquire each of these images?
Yeah, good question. I think for the gills, so it doesn't take that long, but so these images, for example, each one probably took about between 5 to 10 minutes, which is pretty fast for confocal microscopy. Basically, when you're doing confocal microscopy, as I'm sure you know, Tamsin, but for everybody who's not too aware, if you want to get a high resolution image, it usually takes longer. But luckily with this sample, I wasn't looking for, you know, super or very high resolution. I wanted to get an overview of the gill structure so I could have a less magnified image. But yeah, for other samples, it might take you, you know, some people basically spend like overnight or like days taking confocal microscopy images. So luckily with the system, it was pretty fast.
We've got a question that's come through on the chat from iPhone, because that's the ones using it. Hi Medina, could you comment on zebrafish could be used for SARS-CoV research?
Yeah, good question. And I'm sure it will come up in my Viva as well. So I'm definitely using this as practice. Oh, sorry, it's just kind of, anyway. So actually there have been a couple of studies that have looked at whether zebrafish can be used in, yeah, SARS-CoV-2 or COVID research. There's one which uses like a protein from the virus and finds that it stimulates a kind of inflammatory response in zebrafish. But they haven't been able to actually infect zebrafish with the virus yet. So in some ways, it's kind of like what I showed with using these synthetic compounds. You might be able to study certain aspects of the virus, so for example, a particular protein, but it's not clear yet if we'll be able to use it to study the actual infection process.
And we've got a two-part question from Susan. Would there be a change in pH if different reagents are used within the GFP?
Do you mean like what kind of reagents? Do you mean for the antibody binding stage?
just like if you use methyl, orange, phenolphthalein, different reagents to show that can be used in ordinary chemistry? I just wondered, I hope you don't mind me asking as well. Thank you.
Yeah, it's a good question. Not one that I've thought about too much, but I mean, I think different fixative agents can probably have an effect on the protein structure, and the protein structure is what's going to influence the fluorescence for these fluorescent proteins. So I imagine the pH also plays a large role in that, depending on what reagent you're using. So I guess my answer is yes, I imagine the pH does have an effect because it will influence the kind of conformation of the protein structure. I just don't have any example to kind of say exactly, what the effect would be.
And thanks everybody for your questions.
I have one last question, if that's okay.
Oh, yeah.
You mentioned that most of our immune response is after the virus has left the body, and I was wondering how that shows up in zebrafish. Like, how can you detect it?
So yeah, so what I mentioned towards the start was that, so you do have an immune response at the point of when you're infected. It's just that for some infections like flu, the immune response basically just gets out of control and isn't well regulated, whereas in some other infections, basically you have regulatory systems that would dampen down the immune response to allow your body to kind of recover once it's gotten rid of fought off the virus. In zebrafish, so actually something that was another collaboration that we performed was using one of the synthetic compounds that we showed zebrafish respond to. We worked with some researchers from the University of Manchester to see what happens when you have a mutation in one of the genes that's thought to be important for regulating the immune response. And using that, they were able to show that one of the genes, basically if you get rid of it, the zebrafish response basically does become out of control, or it lasts a longer time and it's a larger immune response. So I guess similar in that, and to what I was kind of describing. So yeah, that's kind of one way that you can study this process. Hopefully that makes sense.
Yeah, it does. Thank you.
Okay, great. Thanks. Thanks so much, Madina. I'm just going to pass on to Miranda, but I have to leave for a lecture at 2, but good luck in your defence, Madina.
Thank you so much. Yeah.
As well.
So thank you very much everyone for coming, and for Madina for the amazing talk. I'd also like to especially thank the people who booked through Eventbrite. It's really great to see some external people from outside UCL coming along, because it's a really great opportunity that, although there is that slightly annoying pandemic going on, it means that we can offer our talks online and for free to the public as well. So that's really good. So I'm going to pass back to Chiara, and she's going to filter people into some breakout rooms so that you can socialise and mingle after the talk. No pressure to stay, but just if you'd like to. And once again, thank you very much to Madina.
Thank you so much for having me.